Hey guys, so this video is going to go over plant anatomy, so let's jump straight into it. So there are a few things that you have to be able to do or have to be considered a land plant. First of all, you must be eukaryotic, must have organelles, must have a defined nucleus, all that good stuff. You have to be able to provide your own food, so you have to be autotrophic. You must have a cell wall that's made of cellulose. Other things have cell walls, they're just not made of cellulose. Plants are specifically made of cellulose when it comes to their cell walls. You must go through alternation of generations. That's where you alternate between a gametophyte stage and a sporophyte stage. We covered what that was in the overview of plants video. And then you must have an embryophyte. You must have something to protect your embryo that you spent so much time and energy to create so that your species can continue. So let's go over some basic plant anatomy. You know that your plant is divided into two main regions. You have a root system, which is made up of root tips at the very edge, and then root hairs, these little tiny projections that come off of the main root. Roots purposely anchor plants into your soil. They also absorb dissolved minerals and water, and in some cases they can store food, like these carrots, which are tap roots, and that's pretty much what they're predominantly known for. They store extra food. Now they come in two distinct patterns. The first is called fibrous root systems, which look like this first picture, and it's just a mat of thin roots that kind of spread out continuously. There's no main root. All of them are about the same size. All of them are about the same length. This is a characteristic of monocotyledon plants, things like corn and wheat and rice and barley and stuff like that. Taproots, again, this is an example of a taproot, so think of anything that has a large root. When you pull it up, a carrot, a radish, a beet, anything along those lines, they tend to have one large vertical root, and then they can produce several small lateral branches or roots called root hairs. So like here, and here, and here, and here. All of those would be those small lateral branches or branch roots. And this is a characteristic of dicotyledons or dicots. Picture 3 shows root hairs. And all that root hairs help to do is they increase surface area. So they increase how much absorption that plant can do because you have more surface area to play with. Okay, so we've talked about the root system. The second basic system that a plant is going to have is a shoot system. So that's going to be everything above the ground. It's made up mostly of stems and leaves, and then we have what we call nodes and internodes and buds that come in various shapes and sizes and locations, and then we also have flowers that come off of a special type of bud called flower buds. So let's go through and look a little bit more closely. Now, a lot of plants that we just think of as generic plants are actually modified regions of a plant. So here are some examples of modified shoot systems. Ginger, which we use for seasonings. If you are familiar with me and my, as Kevin likes to call it, my ginger water, this is what the actual ginger looks like. And it's a modified shoot. Then strawberries, which grow in the form of stolons, those are also modified shoots. Here we have potatoes. Yes, a potato is not a root. It's actually a shoot. It's part of the part that would normally grow above ground. This has been adapted to store starch. Then we have onions, or bulbs. If you ever peeled an onion, you'd notice it has all these layers and layers and layers. Well, those are just leaves. They're modified shoots that just fold over themselves again and again and again. So this would be your shoot system, and then right here would be your roots. Potatoes are a part of a family called tubers. That's the name we give to their modified shoots. And ginger are a part of a family we call rhizomes, which is the name we give to their modified shoots. Okay, so we've talked about the root, talked about the shoot. Let's talk about leaves a little bit. Now, leaves are technically a part of the shoot system. They're made up of mesophyll tissue. You remember when we talked about photosynthesis, I showed you what the cross-section of a leaf looks like. and they have veins. And I told you that those veins are their vascular bundles. It's what supplies your plant with its water and its sugar, xylem and phloem. Let's hit the leaves up again. We know that the leaves are the solar panels of a plant. This is the site of photosynthesis. It's where we're going to make energy. It's where we're going to produce glucose. It's also where gas exchange takes place due to those openings called stomata, and it's also where transpiration will take place, where water can evaporate off of the leaf. Now, leaves can be broken down into two big categories, what we call simple leaves versus compound leaves. Simple leaves are just one large leaf on one petiole attached to one auxiliary bud, whereas compound leaves are several individual little leaves, each one's called a leaflet, that come off of an auxiliary bud. So these and these would both be compounds, the difference just being that one petiole gives rise to even more leaflets in this example than it does in this example. Okay, so this would be a simple leaf, this would be a compound leaf, and we call these doubly compound leaves. They're still compound leaves. So let's look at modified leaves. Now, if you've ever looked at ivy, ivy has the ability to climb up like walls and fences and stuff like that, because ivy has, along with peas and a lot of other plants, have modified leaves called tendrils, which pretty much wrap themselves around any sturdy, upright, material, and then allows the plant to pull itself up using those tendrils for support. Here is an example of cacti. Cacti have spines. To a certain extent, we tend to think that they don't have leaves, but those spines are their modified leaves. They're modified because of where these plants live. In that dry and hot desert environment, you don't want to have huge leaves from which you're going to lose a ton of water through transpiration. Then we have succulents. These leaves have evolved to storing excess water, or these plants, have evolved to storing excess water in their leaves. So their leaves tend to be plumper, and they've lost some of those characteristic vein-like structures that we normally associate with leaves. We also have colored leaves. So here's an example of a poinsettia, which is a plant that actually has very small flowers itself. This would be a poinsettia's flowers here. So really small, not very attractive to insect pollinators because they don't have these large pretty petals. Instead, the poinsettia is going to use its very large and colorful leaves to do its attracting for it. So colored leaves are something that we see as an adaptation to plants that generally make either really small flowers or flowers that don't produce a lot of sweet-smelling nectar, nothing to attract those pollinators as needed. All right, so here's that cross-section that I was talking about earlier. Guys, you're really familiar with this. You've seen this a couple of times before, so we're not going to spend a bunch of time on this slide. Okay, now, plant systems, just like animal systems, are interdependent. They both depend on each other. Your roots don't make any food for themselves, so they're going to depend on the sugars made by the leaves because that's where photosynthesis takes place. So photosynthesis happens in the leaf, and sugars get transported from those leaves up to flowers and down to roots. Now, a plant can't live without some kind of supply of water and some basic minerals. That's coming directly from the water and the soil. So your shoots, in turn, are going to depend on your root system to provide them with that water and those dissolved minerals that they're going to need. So the two systems depend on each other. Okay, plants have tissue. I know, it's weird to think of, but they do. Just like how we have tissues, plants have tissue too. They're broken down into three main sections: dermal tissue, ground tissue, and vascular tissue. So your dermal tissue is kind of like the skin of your plant. Its main part is the epidermis. So it's very thin. It's normally a single layer, and it's made of tightly packed cells, so they're really, really close together, and they cover and protect almost the entire part. So if you look at this diagram here, your ground tissue is going to be that white section. So like you can see, it's the smallest set of tissues that your plant has. It's found everywhere, but it's in a really thin layer. Your ground tissue, so that would be the yellow part on this diagram, that makes up the bulk of your plant, okay? And most of it is going to be mesophyll-like in structure. It's where photosynthesis can occur, and it can also provide some storage if necessary. Then in the very center of your plant, you're going to have your vascular tissues. And your vascular tissues are your transport system for both your roots and your shoots. Primarily, they are called xylem and phloem. So that would be the purple regions that you can see on your diagram. So let's go over. Dermal tissues on the outside, that's the white part that you're seeing in these cross sections. The yellow part, which is kind of in the middle, it's the majority of the plant, is your ground tissue. And then the purple part, which is almost dead center in most of these, that is your vascular tissue. Right, so let's talk about the kinds of cells that plants have, because tissues are made up of cells. The first kinds of cells are called parenchyma. I also had a professor that pronounced them parenchyma, so I don't know which one you're familiar with, but I pronounce them parenchyma, so I've heard other people say parenchyma, whatever, as long as you can spell it. So parenchyma, these are typical plant cells, like when you draw or when your teacher draws or when I draw a picture of a plant cell on the board, this is the one that we're normally drawing. It's not very specialized. They tend to be photosynthetic. They can be storage. They're found everywhere. They're found in your leaves, in your stem, in your fruit, in your roots. They're everywhere. The second set are called collenchyma or collenchyma or collenchyma. Collenchyma they're very stocky looking, and they have primary walls that are very uneven but thick, and these guys are mostly for support. So if I could do a little bit of drawing, here's what your parenchyma cells look like. It's just your typical boxy looking plant cell with your nucleus in the middle and your chloroplast and your mitochondrion, and whatever other organelles you have. Very typical looking plant cell. Your collenchyma have more of a stocky box-like look, and they are unevenly thick, so like that almost. Okay, the insides are still going to be the same. But these guys you can tell right away that this is more about support, whereas this is more about function. So I would say this is more of a functioning cell, where this is more of a support cell. The last kind are called sclerenchyma, and these are found only in secondary walls, and they're very thick and very woody. So really, really thick walled. And right away, just based on what they look like, just based on their appearance, you can tell that these guys are all about rigid support. Now, these cells don't really elongate. They stay fairly short and boxy, and they're actually dead when they're at their functional maturity. There's nothing on the inside because they are all about support. All right, so here we go again with actual pictures. So these are your parenchyma cells, like I said, unspecialized, they're thin, they're flexible, they're your typical plant cell. They carry out tons of functions for your plant. It's all of your photosynthesis and your cellular respiration and all of the day-to-day runnings of your plant. Every other kind of cell, sclerenchyma and collenchyma, all develop from parenchyma cells. Like I said before, they're found everywhere, from your flowers all the way down to your roots. Now these are collenchyma. See what I mean about them being unevenly thick? In some areas, like if you look here, this is a really thick region right there. But then here is a really thin region. So they just vary in how thick they are. And it's all primary, what we call primary wall thickness, and it's to help with support. So they help to support your plant, but they allow your plant to grow as well. They don't restrain growth. So these cells are capable of elongating, and they are completely alive even when they're at maturity. So they continually grow, they're continually elongating. If you are a celery lover, you know when you bite into a piece of celery, you tend to have these stringy-like parts that come off on the stalk. All of those are made up of collenchyma cells. And if you think of how rigid and how hard those are, and how they kind of hold your celery stalk up, it kind of goes with the whole support thing. So again, form and function go hand in hand in biology. Okay, here are your sclerenchyma cells. Really thick ridged walls. See? If you look at, let's look at this part of the picture right here. Let me see if I can put some arrows to guide you. Okay, where this dot is in the middle, these little blue or I'm coloring them in now. So these little black dots, that's the actual center of your cell. All that pink stuff that you're seeing around it, that's the actual sclerenchyma cell wall. So really thick, really rigid, really only found in plants that produce secondary wall or wood, lignin. These are the ones that don't elongate. They don't really help your plant to grow. And for the most part, they tend to be dead when they are mature. These are the cells that you're going to find surrounding your xylem vessels and your xylem tracheids, which we will talk a little bit more about when we talk about plant nutrition and plant transport. But this is the part of your vascular bundle that is all about transporting water. They also help to make up fibrous ropes. They help to make up nutshells and seed coats in plants that produce seeds. And when you eat a pear or a lot of other fruit actually, and it's too young, how you get that gritty texture in your mouth, well, that's from those sclerenchyma cells. All right, so let's look at vascular tissue. Your vascular tissue come in two forms, either xylem or phloem. So first we'll look at xylem. Xylem is all about the movement of water and dissolved nutrients, and it transports that water from the roots up to the shoots. These cells are dead at maturity, and not only are they dead, they're also hollow. So think of them as giant straws that run from the very bottom of your plant all the way up to the very top of your plant. The only part of the cell that still remains is the actual cell wall. You need that empty space so that you can move water. Okay, so it's all about transpirational pull. Remember, we did this really, really early on where I showed you like one water vapor moving out of a stomata and pulling on all the other water molecules attached to it because of the cohesiveness and the adhesiveness of water. So that's how water moves in and out. If you look at this picture, it shows you kind of like an x-ray of the inside of these xylem. And all of this white region just tells you that that's dead space. That's empty space. So this is where the water is actually traveling through when they move through these tissues. Okay, phloem. Now your phloem are your food conducting cells. So remember, sugars are only made in the parts of your plant that can do photosynthesis. So the roots aren't making any sugars for themselves at all. So your phloem are in charge of carrying sugars and nutrients throughout your plant. Right? And when you look at them, you can also see they're also fairly hollow. So again, that goes hand in hand with what them, transporting things back and forth. We'll talk more about xylem and phloem when we talk about plant transport, like I said before, in plant nutrition. All right, so here's some other pictures. Okay, so your phloem cells are living cells. They do have a cell membrane, they do have a cytoplasm, and for them it's all about the control of diffusion. Now, while they may have a cell membrane and a cytoplasm, they do lose some of their organelles. They lose their nucleus, they lose their ribosomes, and they lose their vacuole. And if you think about it, these are the three things that are going to take up the majority of room within your plant cell. So they lose these organelles to create more room so that they can transport this liquid food, this sugar that your leaves are making. They're made up of two kinds of cells, sieve tubes, which have pores on either side that allows fluids to move through different cells or between different cells, and then companion cells. Companion cells are found like next to or sandwiching your sieve tubes, and they have a nucleus, and they're connected to the sieve tube, and just like the name suggests, they are companions. They help the sieve tube out. We'll talk about how later on. All right, so we're going to now compare dicots to monocots. Remember, your dicots are things like trees and shrubs, whereas your monocots are things like grasses and lilies, and their arrangements of certain things are very different. So this is a stem of a dicot and a stem of a monocot, and it shows you the arrangement of their tissues, particularly their vascular bundles. So if you look at your dicot, your vascular bundles, I'm going to draw a line, is everywhere this line goes around. So it forms a circle almost on the outside perimeter of your actual plant. The middle part is this region that we call the pith. And your vascular bundles in a dicot are arranged in a very specific way. You have, excuse me, you have your xylem closer to the inside, and then your phloem on the outside, or on the outside of the xylem, I should say. Now, your monocots don't really look like that. I always think of these as looking like monkey faces. Like when you look at these under the microscope and you zoom in, the little bundles look like monkey faces. So this whole thing here, 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 all of these are your xylem vessels in a monocot. So a very different arrangement. And just to say one more thing, it's these xylem bundles in your dicot that give them their annual rings. So, they say if a tree is really old and you cut it in half, you can count how old it is by how many rings you see. Well, part of that's the xylem and the phloem. Okay, now in the root, things look a little bit different. This is a dicot root, and right in the center is where your xylem and your phloem are going to be. So, the part that makes up the cross, because to me it looks like a cross, that's going to be your xylem. And then all the parts are around the cross, like here, and here, and here, and here, and here. Those are your phloem. I have a neat way of remembering which is which, if you're looking at a dicot or a monocot when it comes to these. So if you remind me, I will tell you how I remember it when I see you in class. All right, and then again, here is your monocot in the root. Still a very different arrangement, but this one, it's kind of easier to see because the big circles that you can see here, whoops, and here, all of these big open-looking circles, those are your xylem vessels. And then right behind them there's a smaller arrangement. Right here, and there, and there, and there, and here, and here. Those are your phloem. So we have a ring structure this time in the root for a monocot, and not a ring structure in the root for a dicot. So they look completely different. You just have to remember which is which. And that's it for plant anatomy. Hopefully it was quick and painless, and we'll talk more about it in class. Bye, guys.